So now we will talk about the physiology of the lens. You will find that the same items that we discussed in the cornea, it will be again discussed in the lens. And this is because the cornea and lens are very similar topics because both of them are refractive media, both of them should be kept transparent, and both of them should have some physiological mechanisms to maintain their transparency. So we'll start by the anatomy and the microanatomy of the lens. Then we will study the lens biochemistry. Then we'll pass by the lens nutrition and we'll know what are the different metabolic pathways that are used for supplying lens nutrition. Then we'll talk about what are the mechanisms that maintain the lens transparency and what are the mechanisms that will cause the cataract formation, or what is the cataractogenesis. Then finally, we will end up by what are the accommodation theories. Okay, so talking about the anatomy and the microanatomy, we said that before that the lens is a biconvex structure that is crystalline and lenticular in shape, present in the lenticular fossa between the aqueous anteriorly and the vitreous posteriorly in a saucer-shaped depression. The main function of the lens is not protection like the cornea, but it is mainly visual function. So it will responsible for about 1/3 of the refractive power of the eye, as it focuses the light through the fovea and prevent the damage from ultraviolet radiation. As we said, it will absorb the ultraviolet rays A, so that it will prevent the damage of the retina from the ultraviolet rays. The lens is formed mainly from three structures. From outside to inward, it will be formed by the lens capsule, which is present only anteriorly, and there is no posterior lens capsule. Sorry, the anterior capsule, and there is the anterior epithelium, which is present anteriorly, and there is no posterior lens epithelium. And then will be the lens fibers. The lens fibers will be arranged into a nucleus and into a cortex. Talking about the lens capsule, what are the structure of the lens capsule and how does the structure will serve the function? The capsule is an elastic semi-permeable membrane to which the zonule are attached near the equator, as we know. So this lens capsule will enclose a lens substance. This lens substance is plastic. So here is an important term. What is the difference between elastic and the plastic? So the lens capsule is elastic, while the lens material is plastic. So elastic means that a substance that has a recoil. So when it is stretched, it recoils. And this is a property for a lens capsule. So when it is stretched, like by contraction of the ciliary muscle and the stretch of and by the effect of the zonular fiber, so that it should be elastic, so that it can it stretch and recoil according to the pressure or the force applied to it. And then the lens substance itself, it is plastic. Plastic meaning that something that it molds or it takes the shape of the surrounding structure. So the lens has the ability to take the shape of the surrounding capsule, and this is when the lens accommodate, leading to bulge of the anteroposterior diameter of the lens or a stretch of the lens capsule. Then this will lead to that the lens substance itself will be plastic, so that it will fill the elongated anteroposterior diameter, so that it will be enlarged. The lens is made of, or the capsule is made mainly of type 4 collagen with the glycosaminoglycans made of some outer zonular fibers and inner cuticular fibers that have some functions. What are the functions? These are the main attachment of the zonular fibers, so that it will mold the lens substance during accommodation by the plasticity of the lens substance and elasticity of the lens capsule. Also, it is permeable, so it will be freely permeable to salt, water, dye, especially with the thin posterior capsule. So this permeability will allow nutrients to enter within the lens, so that this capsule will be act as the guard that will allow the glucose, the oxygen, and the nutrients, the amino acids to enter inside the lens. So the lens, this permeability increase with acidity and decrease with age. So as the permeability decrease with age, this means that as we age, the lens will not acquire the necessary nutrients. So that this was what will be one of the mechanisms for cataract formation. So as we go, you can count what are the numerous number of the mechanisms that are responsible for cataract formation. So as we said in the cornea, that every mechanism which is the one is disturbed, it will lead to impairment of the corneal dehydration or impairment for the corneal transparency. Again, here in the lens, as we go, there will be several mechanisms. When they are disturbed, this will lead to impairment for the lens transparency and leading to cataract formation. After the capsule, beneath the capsule, there will be lens epithelium, and the lens epithelium is only anterior. As we know from the embryology and from the anatomy, the posterior lens epithelium was consumed during the embryonic life by formation of the primary lens fibers. So we only have anterior lens epithelium that have a difference in size and shape. So they are cubical in the center, and they become more elongated at the equatorial region as they will divide and elongate, forming the lens fiber afterwards. The anterior lens epithelium is a metabolically active layer. So here we need the ATP. So we need here ATP. As this in this layer there will be a sodium potassium ATPase pump again. As this pump will pump excess water and salts that are coming from the aqueous. Also, this layer contains DNA and RNA for protein and lipid synthesis and supply the ATP for energy production by the lens. So here is the main source of energy that will supply energy to the lens for energy production that is important for maintaining transparency, as it will pump the fluid. And also it will allow the nutrients to enter within the lens in order for protein synthesis and lipid synthesis. So damage of the epithelium, if there is damage to epithelium, this will lead to accumulation of water in the extracellular spaces as the pump is not working anymore. So this will lead to cataract formation. So what are the clinical features or what are the clinical significance of lens anterior or the anterior lens epithelium disorders? Sometimes there can be some anterior subcapsular cataract. So this is anterior subcapsular cataract occurs due to metaplasia occurs in the central zone. And this cataract usually is not visually significant and usually doesn't need interference, and it can be removed later on if the patient developed any kind of cataract that is visually significant. It can be just removed with the capsular access. Glaucomflecken is a kind of necrosis of the anterior lens epithelium that occurs due to ischemia caused by an acute attack of angle closure glaucoma. That's called glaucomflecken. So anterior epithelial necrosis. PCO or posterior capsular opacification, meaning that there is migration of equatorial cells toward the posterior capsule, as you can see here. These are called the PCO. So this is just the residual cortical fibers that are, or equatorial cells that will migrate to reach the posterior epithelium or the posterior capsule, causing these opacification. Then after talking about the lens capsule and the lens epithelium, we have what's called lens fibers. So the lens fibers here it is the same, it is more or less like the collagen fibers present in the cornea. So these are the main content which is responsible for the lens transparency and responsible for the lens action. So this will be responsible for convergence of the light rays, and this will be responsible for refractive power of the eye. So we have here the capsule and then the lens epithelium, and the lens epithelium will enclose the fibrils. So fibrils are divided or are hexagonal in cross section. This is asked in the MCQ. As you can see here, it is hexagonal. The primary lens fibers, as we said, will be consumed in the embryonic life by formation of the posterior, or it's formed by the posterior epithelium during the embryonic life. Then later on, it will be constantly formed by elongation of the equatorial lens epithelium from germinative cells. So as they enlarge, they will form the nucleus, and the old fibers will form the nucleus, and the recent fibers will form the cortex. The nucleus will be formed of several nuclei that are developing throughout the years of life. So starting first by the embryonic nucleus, which will be developing in the early embryonic months. So it will be in the first 2 months of embryonic life. Then the fetal nucleus will be developing until the last month of gestation. Then infantile nucleus that will be formed later on until the adulthood. Then the adult nucleus will be formed later on. The cortex will be made from the superficial, or will be making the superficial part, so that these are the youngest lens fibers. So as the lens fibers are laid down from the equator, these making the cortex, and the cortex will compress the older fiber inside toward the lens nucleus. It should be mentioned also, optically speaking, that the lens is like this. It's formed like this. So it's formed of central nucleus and then the peripheral cortex. So the peripheral cortex can be considered like a minus lens, while the nucleus can be considered like a plus lens. After having a quick summary of the lens anatomy, let's now discuss the lens biochemistry. So lens biochemistry meaning that what are the contents of the lens? So here again, the numbers are very important. So the water here making about 65% of the lens. And this can be considered relatively low. And this is a relatively low water content in relation to other tissues within the body. This is because to maintain the lens transparency. In contrast to water, or to the relatively low content of water, there is a relatively high content of proteins. So proteins, we can say that the proteins making about a third of the lens content, or 34% of the lens content. This is one of the most, if it is not the most highest protein ratio within the body. So this is the highest protein ratio of any tissue within the body. So this is very, very important and will be discussed in much more details. Finally, there is a 1%. This 1% will account for lipids and carbohydrates and salts. So let's start with water. So water makes about 65% of the lens weight. As I told you, it is relatively low compared to the other tissues, and this is important to maintain the refractive index and to maintain the lens transparency. Lens dehydration, or this relatively low, is caused by the active sodium pump. So same as the cornea, the sodium potassium pump plays a major role here to maintain the lens transparency by pumping out the excess water. The cortex, since the cortex is more located outside, so it is more located near the vitreous and near the aqueous, that's why it is more hydrated than the nucleus. And as we get older, the water content decreases by age, leading to decreasing deformability, leading to cataract. So this is one of the mechanisms, again, formation of cataract. As we get older, the water content decreases, so that the water content is also important for maintaining transparency within the limited amount, or within the relatively low amount. If this decreased more than this by age, then this will lead to that the lens lose its flexibility, so that the lens will not be able to contract and relax, and the lens proteins will be overriding over each other, and this will be one of the mechanisms responsible for cataract formation. There is a relation between lens and diabetes, meaning that if there is hyperglycemia In case of hyperglycemia after heavy meal, then the blood has high osmotic pressure. So this hyperosmolarity will be transferred to the vitreous, so that this will lead to hydration of the cortex. So this hydration of the cortex will cause that this cortex will decrease in refractive index. And since that the cortex acts as a minus lens, if the minus lens decrease in refractive index, this will lead to myopia. So this will lead to myopic shift. So again, hyperglycemia can cause myopic shift. How? This is by hyperosmolarity. Hyperosmolarity will cause hydration of the vitreous. This hydration of the cortex. The cortex acts as a minus lens. So if the minus lens is weakened by decreasing refractive index, then this will lead to myopic shift. The reverse occur in case of hypoglycemia, like if the patient take an excess insulin dose. So if there is hypoglycemia, there will be hypoosmolarity, and this hypoosmolarity will cause dehydration of the cortex, as since that the vitreous is hypoosmolar, it will draw out the fluid from the cortex into the vitreous, so that this will lead to increasing the refractive index, as the cortex will be very stiff. And since the cortex acts as a minus lens, with increasing the refractive index, this will lead to hyperopia. So hypoglycemia causes hyperopia. Hyperglycemia will cause myopia. Let's talk about a very, very important issue in the lens, which is lens proteins. We need to know what are the lens proteins, which one of them are responsible for maintaining lens transparency, and which one of them are not related to the lens transparency. What are the different types, which one of them are larger, which one are smaller, which one are numerous, which one are few, and what are the other functions of these lens proteins. So lens proteins can be broadly divided into water-soluble lens proteins, which accounts for 85% of the lens proteins, and the water-insoluble. The water-insoluble will be making about 12.5 of the lens proteins. The other name for this water-insoluble proteins is called the albuminoids. The albuminoids. So the water-soluble lens proteins can be further divided into alpha crystalline, beta crystalline, and the gamma crystalline. And by saying crystalline, so these are what are making the lens crystalline. So the water-soluble lens proteins are the proteins responsible for the lens transparency. And water-insoluble lens proteins have other functions and can be further subdivided into urea-soluble and urea-insoluble. This is very important. Let's start by the crystallines. We have the alpha and the beta and the gamma crystallines. So what are the crystallines? They are forming about 90% of the lens proteins. So they have the refractive function. So they are the main lens proteins. They are the main issue or the main responsible for the lens transparency and the lens function of refraction. They are able to change their shape during cell differentiation. They are stress resistant and they have oxidative properties. We will discuss this later on. And they have what's called the chaperone-like activity or chaperone-like function. This is a function of specifically for alpha crystalline. And the alpha crystalline do what's called stabilization of the unfolded proteins, and they prevent their aggregation so that they keep lens transparency. So if there is aggregation or any if there is protein that have been unfolded, like so the proteins are all kept in regular and specific configuration. If they are unfolded, then this alpha crystalline will will stabilize this unfolding this lens protein, which if they are kept unfolded, they will undergo aggregation, so that this will cause the lens to be not transparent. So how can these crystallines change in order with age in order to to share in the formation of cataract? So as the lens change, as we age, this soluble alpha crystalline get gradually converted into insoluble albuminoids, leading to cataract. So one of the major mechanisms responsible for cataract formation is that the crystallines are or the water soluble proteins are changing into water insoluble proteins so that they become they form a cataract. We have three major types of crystalline. We have the alpha and the beta and the gamma. This is very important to know that the ratio it is the 35% for the alpha, 55% for the beta, making it the most numerous, and 1.5% of the gamma, making it the least numerous. So what is the specific about alpha? It is the largest. This is for MCQ. So alpha crystalline is the largest, beta crystalline is the most numerous, gamma crystalline is the least numerous or the most few. So here is the lens, and here what is called the damage or changes that occur to the crystalline. These are in the unfolded. This is the folded configuration, and if they are unfolded, then the alpha crystalline act as what is called the chaperone-like activity. This is also asked in the MCQ, so that they will stabilize these unfolded proteins and prevent their aggregations. If they aggregate like formation, if they aggregate like this, they will make the lens opaque. The second type of the lens proteins are called the albuminoids. So the albuminoids are divided into two types. There will be urea soluble and the urea insoluble. The urea soluble are making the most of the cytoskeletal proteins. This is the major function of the urea soluble. So they are not sharing in the formation of refractive power of the lens or not maintaining the transparency of the lens, but they make the skeleton of the lens. So they make the skeleton of the proteins, or they form cytoskeletal proteins that will make the framework of the lens. The urea insoluble are making or form the most lens fiber membrane protein, including the major intrinsic protein. This is asked in the MCQ. So one of the membrane proteins or the lens fiber membrane proteins is called a major intrinsic protein, which is a urea insoluble. So this is just a protein that is making the lens membrane or the cell membrane of the lens fiber. This is made by the urea insoluble protein. So what are the functions of the lens proteins? Well, the lens proteins will stabilize the lens fibers, as we said, like the chaperone activities, and prevent their separation during accommodation. Also, they are responsible for maintaining the lens transparency by the alpha crystallines and the beta crystallines and the gamma crystallines. There are some other proteins that are present in minor amounts in the lens, such as phosphoproteins and the glycoproteins and lipoproteins. The cortex itself is rich in crystallines, and the nucleus is more in the albuminoid. So if you are asked which of one is more the crystalline or albuminoid, the crystalline is more in the cortex, while the nucleus is more or more rich in albuminoid. And the lens protein are immunologically privileged sites. So this means that the lens proteins are not identified by the immune system, as they are sequestrated within the lens capsule since the embryonic life, so that the immune system doesn't recognize the lens proteins. This means that once the lens capsule is opened and the lens become or the lens protein are arising or emerging outside the lens, then the immune system will identify them as a foreign body and will start to attack them. And this is what is called the lens-based uveitis, or can cause what's called the phacoantigenic or phacoanaphylactic uveitis. Then So we talk about the water, then the protein, and let's now talk about the minor elements present in the lens, such as carbohydrates. So carbohydrates present in the lens in different forms. The most important will be the glucose, which is coming from the aqueous humor. It has a concentration between 20 to 120 milligram percent, and the glucose in the lens is 1/10 of its concentration in the aqueous. So here is a very important question in the MCQ. They tend to ask you, well, what is the concentration of the glucose in response to the aqueous? So you may think, oh, the glucose is very important source of energy in the lens, so it must be very, very much in the lens more than the aqueous. But the surprise here, although the glucose is needed in greater amount in lens, but it is consumed very rapidly. So the amount of free glucose within the lens is less than or about 1/10 of its concentration within the aqueous, because it is just rapidly metabolized. Fructose can also be one of the carbohydrates that are produced from the glucose, as we will see later on. The glycogen is very high in the lens and present in the nucleus, and the other carbohydrates can be also present, like inositol and the sorbitol. About the plasma lipids, we have the cholesterol, so it will make about 50% to 60%, which will be the major lens lipid. Other lipids such as phospholipid and sphingomyelin and glycolipids are also present. So their functions are mainly to stabilize and to form the lens membrane. So lens membrane is made of lipids. If you remember from our undergraduate years, the lens lipids like sphingolipids are sharing the formation of the cell membrane. So this is one of the main functions of the lipids here. Also can be used or can be associated with the lens epithelial cell division. Many electrolytes such as cations and anions are present in the lens, such as potassium. So potassium is the main cation present in the lens. And the sodium will be about 10 to 50% of the potassium concentration. The calcium here is the striking one, as it has the lowest concentration in all tissues, as it is 50 times less than the aqueous. So it is only required in very minimal amount to maintain the cell membrane permeability. So it's important to maintain the conduction through the cell membrane, but if it increases above its limits, then it will lead to cataract. So it can cause cortical cataract. That's why calcium shouldn't be increasing in the lens. So these are the main cations. Main anions are the chloride, the bicarbonate, the phosphate, and the sulfates are also present within the lens. A very, very important molecule or substance present in the lens is called the glutathione. So what is the glutathione? Glutathione is a tripeptide compound that is, since it is very important, they make about 3.5 to 5.5 milligram per gram of net weight of the lens. So where does or what is the source of this glutathione? It comes from the lens epithelium. So lens epithelium contains high levels of glutathione, and about 95% of this glutathione are in reduced state. Why? This is because it will act as an antioxidant or reducing agent, as we will see later on. So this will combine, or the glutathione is the main antioxidant of the lens. So it will combine the oxygen to decrease the or to minimize the damage or the damaging effect of the oxygen-free radicals. Permeability and the transport mechanisms can be divided into two main mechanisms. The active transport using the ATP, so this is by the sodium-potassium ATPase pump, and this will be the mechanisms responsible for transport of amino acids, so that we need the amino acids to produce the lens protein, so we need it in excess amount. Potassium, so we need to activate the sodium potassium ATPase pump, and the other amino acids such as taurine, the inositol, can be also extruded from the lens by the sodium potassium ATPase pump if it is accumulating inside the lens, and extrusion of the sodium by the sodium potassium ATPase pump. The passive transport can be also the mechanism for transport in case of water and ions, and the removal of the waste products from metabolism, such as the lactic acid formed by the metabolism or the anaerobic metabolism of the glucose and the carbon dioxide. As we said, the lens capsule is permeable to low molecular weight compounds and restrict the larger colloidals. There is what's called the cation pump, which is functioning at the anterior lens epithelium. So with the help of the ATP, the sodium is actively removed from the lens and uptake of potassium occurs. So as the sodium is removed from the lens, this will lead to a removal of water. And this process of active transport will stimulate what is called the passive diffusion. So passive diffusion of water occurs after removing of the sodium. Lens also can be considered as an osmometer. So it can be considered as a single giant cells that swells up in hypertonic media. So since that it is surrounded by fluids anteriorly and posteriorly, so there is a concentration for fluid within the lens, and there will be a concentration of the molecules within the lens, so that it has an osmotic pressure. If there is increasing in the sodium and the potassium, so that it will increasing the molecule, this will increase the osmolarity. And since there is increase in the osmolarity, the lens will absorb water and will draw water from the aqueous and vitreous, and this will lead to loss of the lens transparency. So here you can see this is the lens and this is the aqueous. This is a very fantastic figure. You can see that although the water content in the aqueous is 99%, it is about 66% on the lens. So the lens has relatively low water content to maintain the transparency. The sodium potassium ATPase pump will cause extrusion of the sodium, so the sodium will be few. Here in the lens it will be 20 and 144 in the aqueous, which is the same concentration like the plasma. And this is so if you find that there is a large difference in between two areas around or across a semi-permeable membrane, so this means that this is removed by, or this is an active pump here. So this is against concentration gradient. The same here with the potassium. As you can see, the potassium is actively introduced inside the lens, so that it will be only 4.5 in the aqueous and it is 125 in the lens. The chloride pass into the lens by the diffusion, and the glucose, although it is needed in much more amount, but it is less in the lens than in the aqueous, because it is rapidly absorbed and it is rapidly metabolized. Lactic acid, as a result of waste products, it will be diffused through the semi-permeable membrane of the lens capsule into the aqueous. Glutathione is 0 in the aqueous and 12 in the lens. It is also it is brought from the aqueous and it is synthesized in the lens, because it is very important in maintaining lens transparency, as it is the main antioxidant of the lens. Some sugar like inositol, it will enter by active transport. Amino acid will also be entered by active transport to maintain synthesis of lens proteins, and the protein content will be about 33%, as we said, so 33 to 34. So this is the same figure illustrating the different mechanisms of diffusion and active transport of different molecules and proteins and water between the cells and the aqueous. So the active transport is mainly for amino acids, sodium, potassium, inositol, and 90% of the energy in the form of ATP is utilized here. However, the passive transport is mainly for water, for ions like chloride, for waste products like the lactic acid. Transport of amino acids is by both pump mechanism and a mechanism called the leak mechanism. The glucose transport is caused by the simple diffusion or another mechanism called facilitated diffusion. This is important and asked in the MCQ. So one of the mechanisms for transport of glucose called the facilitated diffusion. So speaking about the lens, what are the contents that enter the lens and what are the exit from the lens? Here we come to the lens nutrition and the metabolism. So the lens is like the cornea. It is a transparent structure and doesn't have any vascular blood supply, and that's why it needs its nutrients from it, or it obtain their nutrients from the surrounding structures. So it takes the nutrients from the aqueous and the vitreous, and and sometimes some of these contents or nutrients are needed in much larger amounts. That's why it needs a source of energy for the sodium potassium ATP pump. So why we do need ATP? For transport of ions and amino acids, for maintenance of the lens dehydration, and for continuous protein synthesis, and also for glutathione synthesis. The major site for the active pump or the sodium potassium ATPase pump, as we said, it will be the anterior lens epithelium, and the source of energy will be coming from the aqueous humor, or source of the nutrients will be coming from the aqueous humor. So what are the different pathways of glucose metabolism in the lens? So what are the sources of energy? Well, the energy here in the lens is mainly dependent on the glucose, or it is only on glucose. So we need to metabolize glucose in order to provide energy for the lens that will maintain the lens transparency. So how can we get, or how does the lens obtain this glucose? This can be done by diffusion, simple diffusion. And if it is still not enough, then the lens will do facilitated diffusion to allow a fast entry of the glucose to the lens. Then the glucose is rapidly metabolized via glycolysis. That's why the concentration is less than 1/10 per its concentration in the aqueous. So what are the metabolic pathways? We mentioned them in the cornea, but let's now discuss them in more details, because here numbers are important. So we need to know what are the main metabolic pathways and how much energy production by the lens accounts for each pathway. For the anaerobic metabolism, it accounts for 85% of the glucose metabolized by the lens, as it provides 70% of the energy production by the lens. So again, the anaerobic metabolism will about 85% of the glucose metabolized by the lens will be metabolized by the anaerobic metabolism without the presence of oxygen. And this will provide more than 70% for energy of lens. You may wonder why, but this is because that the only part of the lens that has a good exposure to oxygen will be the anterior lens epithelium. So what about the other parts of the lens? So they will not be functioning, they will not do any glucose metabolism? The answer, of course, no. But they will metabolize glucose by the anaerobic metabolism. And you can find that this metabolism doesn't it will share in a great part by of the total energy required by the lens, as it will provide more than 70%. The drawback of the anaerobic metabolism, two things. First thing, that it will result in low amount of energy, as 1 mole of glucose will be metabolized to only 2 moles of ATP. The second problem, that there will be a lactate or lactic acid generated from the anaerobic metabolism. So the lactic acid can be either undergo two pathways of metabolism, either further metabolism through the Krebs cycle if there is oxygen have been present, or it will diffuse from the lens to aqueous. So this will be the first metabolism, or the first mechanism. The second mechanism is the aerobic. So the aerobic metabolism is meaning that the glucose is metabolized through the Krebs cycle in order to give about 38 mole of ATP. So this only occur in the lens epithelium since it will be exposed to oxygen, and the advantage of this cycle or this pathway, that only 1 mole of glucose will give 38 moles of ATP. So a huge amount of energy is produced through this pathway. But unfortunately, only 3% of the glucose metabolized by this pathway, because it is only limited to the epithelium. However, this 3% of the glucose can produce 20% of the total ATP needs. So did you understand what is the ratio? So the anaerobic metabolism, about 85, or let's say it is 80% of the glucose metabolized by the lens. So this is provide more than 70% of energy of the lens. But although that only 3% of the glucose is metabolized through the aerobic, but since this 3%, if every 1 mole of glucose will produce 38 mole of ATP, then this only 3% can produce as large as 20% of the total energy requirements of the lens. Okay, so are there any pathway left? Yes, we have the hexose monophosphate shunt. The hexose monophosphate shunt is accounting for 5% of the glucose metabolism by the lens, and one of the main advantages of this pathway, that it will be an important source for NADPH. So this NADPH, or this compound, will be required for other metabolic pathways such as sorbitol pathway, in order to metabolize sorbitol if it accumulates within the lens, and the glutathione reductase, which will be the enzyme that will be responsible for formation of the glutathione, or reduce the glutathione, which will be the main antioxidant of the lens. So the main advantage of the hexose monophosphate shunt, that it is not only present, that doesn't only support the energy production of the lens, but it will be providing an important structure, which is the NADPH, which is required for other functions. There is a new pathway in the lens that is not present in the cornea, which is called the sorbitol pathway. This sorbitol pathway will account for 5% of the glucose metabolized by the lens. And as the sorbitol accumulate within the cells of the lens, it will increase the osmotic pressure. This increasing of the osmotic pressure within the lens will cause watery entry, or water entry inside the lens, and lens swelling, and ultimate loss of the lens transparency. That's why we need to get rid of this sorbitol, and this here comes the value of the NADPH. So the sorbitol pathway, so it is a sugar. It is another sugar than the glucose. So its metabolism will cause some energy production, but if it is accumulated within the lens, it will cause increasing the water. So here is the major pathway of glucose metabolism. In case of glucose, and if there is oxygen, then the glucose will form the glucose 6-phosphate. And this glucose 6-phosphate now will enter what is called the will form the pyruvic acid, and this pyruvic acid, if there is oxygen, will form the acetyl-CoA and will enter the Krebs cycle, forming about which will account for 3% only for the glucose metabolized by the lens. But if there is no oxygen, then under the effect of lactate dehydrogenase enzyme, it will form the lactic acid or the lactate. Also, the glucose can enter other metabolic pathway. It can will enter through the hexose monophosphate shunt or pentose monophosphate shunt, which will form about 5 to 10% of the glucose or the energy production, or it can be transformed to the sorbitol using the enzyme aldose reductase, which will be entering through the sorbitol pathway. So here is the glucose, and then it will be transferred or be transformed into the glucose 6-phosphate. This is called the rate limiting step. So this is asked in the MCQ. The glucose is transformed into glucose 6-phosphate via or by the aid of catalyzed by the hexokinase enzyme. This is called the rate limiting enzyme. And so these are the illustration again of different metabolic pathways. And this table will summarize the pathways like glycolysis, Krebs cycle, hexose monophosphate shunt, and finally the sorbitol pathway and the energy production for each one of them. So glycolysis will form 2 ATPs, Krebs cycle about 36 ATPs. The hexose monophosphate shunt will not form ATPs, but it will be an important source for NADPH, and it will 15% or 10% of the glucose metabolized by this pathway. Sorbitol pathway will form 2 ATPs only. So we spoke about the oxidative damage or the oxygen-free radicals. What is meant by the oxidative damage? There are some metabolic activities that occurs within the cornea and within the mitochondria, and this mitochondrial activity, or as we know that the mitochondria is the house of energy within the cell. So since the mitochondria is a very metabolic, a very active organelle, then it will produce some products, and the metabolic process it will generate some harmful products are called oxygen-free radicals. So the problem of this oxygen-free radicals that it will destroy the cell membrane lipids and the cytoplasmic proteins, and this is one of the reasons that will cause loss of the cell, that it has been destroyed by its own metabolic products. So if there is a hydrogen peroxide, the hydrogen peroxide produced in mitochondria, if combined with the ferrous iron, this will release the damaging free radicals. This is called the Fenton reaction. So if the hydrogen peroxide, again, produced in the mitochondria, if it is combined with the ferrous iron, this can cause releasing the free radicals, and this is called the Fenton reaction, and we'll see about this later on. So this is one of the major slides in this lecture, which is what are the antioxidants inside the lens, and how does they prevent the oxidative damage of the oxygen-free radicals. The first of all, or the first antioxidant, is the low oxygen tension in the aqueous, so that the lens is not exposed to large or high tension of the oxygen, so it will not cause damage by this high tension to the lens. The second thing is high concentration of the transferrin. So the transferrin will combine the ferrous iron. Why it is important to combine the ferrous iron? Again, to prevent the Fenton reaction. So the Fenton reaction, when the free iron is combined with the hydrogen peroxide, this will lead to releasing free radicals. So if this ferrous iron is combined with the transferrin, then it will not bind with the hydrogen peroxide, so this Fenton reaction will not be formed. Then we have the two main antioxidants in the lens. The first one, which is the most important, is called the glutathione. So the glutathione is the main antioxidant of the lens. Its concentration is about 1,000 times more than the aqueous. This is important. It comes mainly from the aqueous and the lens epithelium and superficial cells, and then will be the lens epithelium and the superficial cells. So, are the deeper cells will be deprived from the glutathione? They will require or they will acquire the glutathione, the reduced glutathione, by diffusion from the superficial cells. But the superficial cells will have more glutathione or more concentration of glutathione than the deeper one. The high concentration of the reduced glutathione in the lens will maintain the lens proteins in the reduced state, so that this will ensure that there will be integrity of the cell membrane. So the glutathione maintain its function as an antioxidant in the lens by presence in the reduced state. By meaning that it is present in the reduced state, it is combined with hydrogen. So when it is combined with hydrogen and it finds that there is some oxygen-free radicals or there is excess oxygen, it will combine with this, leading to formation of H2O, so that we will prevent the damage of this free oxygen to the lens. So that it will prevent the damage of this oxygen to the cell membrane. The problem is that glutathione will decrease with age as anything else, so this will be one mechanism responsible for cataract formation. The second thing, or the second antioxidant in the lens, is called by the ascorbic acid. And here is a crucial issue here. It is not formed by the lens. So the glutathione is synthesized by the lens, and its concentration become more than its aqueous by 1000 times. But the ascorbic acid or the vitamin C is not made by the lens. It is acquired from the aqueous and from the vitreous. So it is present in the high content in the aqueous. It will be about 20% of times of the blood, so that it will be react with the free radicals to form the dihydroascorbate. And the lens epithelium so this is the main function of the ascorbic acid. It is acquired from the vitreous and the aqueous, which is present in high concentration, so that it will be react with the free radicals, cause the dihydroascorbate. Also, lens epithelium has some enzyme like catalase that will convert the hydrogen peroxide, which is one of the free radicals that cause oxidative damage. So it will convert the H2O2 into water. So here is the lens. So the lens, since the lens has a low oxygen, so the ascorbate binds with the oxygen, forming the dihydroascorbate and H2O. If there is less ascorbate or there is high oxygen tension, then it will enter through the lens and it will cause cataractogenesis. Okay, so to summarize, what are the mechanisms responsible for the lens transparency? Thin epithelium, regular arrangement of the lens fibers, as we said in the cornea as well, little cellular organelles, little extracellular space, orderly or regular arrangement of the lens proteins, and the prevention of their aggregations, relative dehydration by low water content, semi-permeable character of the lens capsule, so it will allow diffusion of the nutrients and it allow diffusion of the waste products, the avascularity, and this is the new thing increased over the cornea, which is the antioxidant. So if these mechanisms are disturbed, what will lead? This will lead to age-related changes. So how can cataract is formed, or what are the changes that can occur that will lead to cataract formation? So if the water content increase, this will lead to cataract. Sodium, if it is increased, it will lead to cataract. Increasing calcium, as we said, will cause cataract. Insoluble proteins, if it increase, it will cause cataract. What are the factors that may decrease the potassium? The enzymes such as the catalase, the glutathione and the ascorbic acid as they are the main antioxidants, and the ATP. So this slide just to summarize, if you are confused in the exam what increase and what decrease, so you know what are the factors that when increasing cause decreasing transparency, and what are the factors that when decrease it will cause decreased transparency. So by saying decreasing transparency, we just mean cataractogenesis. So cataract meaning that there is a disturbance in the transparency of the lens, which will lead to opacification. The age-related cataract is the commonest type of cataract, which occur mainly above age of 50-year-old, usually bilateral, with many factors contributing to it. There are different types of cataract. This now discuss what are these types of cataract and what is the cause behind each one of them. The first one is called the nuclear cataract. In case of nuclear cataract, you find that the nucleus is acquiring a yellowish discoloration, so the becoming have yellowish hue, and this is because of increase a pigment called urochrome. This is the same pigment which is present in urine. So this urochrome present in the lens will absorb the light so that it will make the lens appear yellowish, and if it increases, the lens appear more orange or more brown. So what are the mechanisms behind the presence of the nuclear cataract? So the nuclear cataract occur mainly due to poor diffusion of the glutathione toward the center. So as we get older, then the amount of glutathione is decreasing, and since the glutathione is made mainly by the anterior lens epithelium, so it will reach the deeper fibers by diffusion from the superficial fibers, so that the nucleus present more deeply and it doesn't acquire enough glutathione. That's why it will opacify. Also, the absorption of the ultraviolet rays, we said that the lens will absorb with the ultraviolet rays A. That's why it will one of mechanisms responsible for nuclear cataract, and decreasing the water content, causing dehydration and the compactness of the nucleus, or increase in the water insoluble proteins like the urea soluble proteins, causing more light scattering. So here are the grades of the nuclear cataract. It starts with nuclear sclerosis and nuclear 1 and 2 and 3 and 4 and 5, causing brown or brunescent cataract. The cortical cataract, which occur more in the anterior or posterior or equatorial cortex, appears as spokes. These are spokes of whitish opacities, usually present peripherally, as you can see here, which are usually due to disturbance of the ionic pump in the cell membrane, causing accumulation of calcium. So remember that the cortical is calcium. So calcium disturbance will lead to, or accumulation of the intracellular calcium will cause increase in the cortical cataract formation, as it will lead to a problem with protein aggregation and the plasma membrane disturbance. So sodium and the calcium content increase of cortical cataract, and the potassium content will decrease, as we said before. The subcapsular cataract can be either anterior subcapsular cataract, as we said, which lies beneath the anterior lens capsule due to fibrous metaplasia, or can be posterior subcapsular cataract, which will be lying behind, or sorry, in front of the posterior capsule. It will appear as granular opacities, as you can see, or vacuolations that are very visually significant, especially if they are present near the center because of involvement of or as they are close to the nodal point, and they are caused by clusters of swollen cells. Radiation also can cause cataract because the radiation can be absorbed by the lens. And there are three mechanisms of the radiation that can cause cataract, as it cause leakage of potassium, inositol, and the glutathione, can cause incorporation or decreased incorporation of amino acids into the proteins, and arrest of cells entering mitosis and epithelial damage as any radiation exposure to the body. So infrared radiation is totally absorbed by the albuminoid proteins. This is important. The infrared radiation is absorbed by the albuminoid proteins and usually it doesn't cause any harm except if it is very high intensity. Then it will lead what's called the true exfoliation syndrome. True exfoliation meaning that the anterior lens capsule will be exfoliated or damaged and removed by itself, and this happens in case of glass blowers. So those who are doing the glass blowers, they wear spectacles to protect their eyes from the infrared, which can cause damage to the lens, causing the true exfoliation, or causing the retina, causing the solar maculopathy, as we said before. So what is the effect of the infrared on the lens? It causes what's called true exfoliation. So there is a superficial zonular lamella of the capsule will split off from the deeper layer and due to exposure of the infrared radiation. This shouldn't be confused by a disease, a well-known disease called the pseudoexfoliation, in which there is deposition of the fibrogranular material all over the body organs, including the lens, and it will making an appearance of a pseudocapsule in front of the lens. So this is a pseudoexfoliation, not a true exfoliation caused by the infrared rays. Also, the ultraviolet A is absorbed by the lens, as we said before. That's why if the patient is removing lens, he will see a bluish discoloration unless the intraocular lens implanted have an ultraviolet protection. The sugar can also cause cataract. This can be through two mechanisms, either the increase of the blood glucose, hyperglycemia, or the galactosemia, or increasing the galactose. So let's say, for example, what are the mechanisms responsible for cataract formation in hyperglycemia? So if there's increased glucose in the aqueous, this will be absorbed by the lens. So glucose will enter through the lens. Then this glucose will enter through the sorbitol pathway. Then this will lead to accumulation of sorbitol. And then the fructose will increase. This sorbitol will be metabolized to fructose, and this fructose will increase the osmolarity of the lens. So the lens will be having high osmotic pressure. This high osmotic pressure will imbibe water from the surrounding vitreous and aqueous, causing more entry of the water into the lens fibers, causing rupture of the of membrane of lens proteins and loss of potassium and amino acids. So this is the one mechanism. The second mechanism that the glucose will be formed or will be transferred. The increasing glucose in the lens will lead to again formation of sorbitol, and this sorbitol pathway through the aldose reductase enzyme will consume the NADPH. So we said that the NADPH is an important structure here. So this pathway will cause reduction of the NADPH into the NADP, so that we don't have a reduced NADP or we don't have NADPH, so that the sorbitol will cause NADPH depletion or decrease, so that the glutathione, the reduced glutathione is depleted, because the reduced glutathione is dependent on the presence of the NADPH. So the main antioxidant of the eye or of the lens is not functioning well, because it is not reduced, because there is no NADPH, because the glucose to sorbitol pathway has consumed the all NADPH. So what are the types of cataract formed by the diabetes? Diabetes, in case of juvenile cataract, it can cause what's called the snowflake cataract, which appear as multiple whitish opacities beneath the anterior or posterior lens capsule, or can cause, in adults, it can cause the cortical or subcapsular cataract, which is progressing more rapidly. And how does the galactosemia can cause cataract? So if the galactose increase in the aqueous, this will be absorbed by the lens. So it will increase galactose in the lens, then it will enter the sorbitol pathway. So by sorbitol pathway, it will be transformed or will be metabolized to a component called alcohol galactitol or dulcitol. This is important because the lens cannot get rid of this product. or the lens doesn't have the necessary enzymes to metabolize this product. So it cannot metabolize or even cannot diffuse out. So it is trapped within the lens. And this will increase again the osmotic pressure within the lens, so it will pull out into the lens, pull water into the lens, causing rupture of membranes and loss of potassium and loss of amino acids. So here is the picture or the clinical appearance of the galactosemia, or presence of accumulation of the galactose within the lens. This will cause what's called the oil droplet cataract. So there will be appearance of a central oil droplet-like opacity within the lens. Finally, the accommodation. So by speaking about the lens, we cannot omit the accommodation and what are the mechanisms responsible for it. So what is accommodation? We know from optics and anatomy that the accommodation is a mechanism by which the lens increases its curvature so that it will acquire more power, it will acquire more bulge to see the near target, so that it will increase its refractive power to see from distance to near. So this will be as a result by changing the lens shape due to the effect of the zonular fiber under the effect of the ciliary muscle. So the accommodation is under the control of ciliary muscle, which exerts its function on the zonule. Then this zonule will act on the lens. So how can the accommodation occur? There is a 3 C's. So this will be accompanied by constriction of the pupil, convergence of the eyes, and increase in the anterior curvature of the lens. So we need to know what are the theories of accommodation. So back to theories again. So since we say theories, so we don't have an actual explanation. The first one is called the relaxation theory, that is suggested by Helmholtz. So Helmholtz theory, or what's called the capsular theory, suggests that when the eye is at rest, then the ciliary muscle here is relaxed. And when the ciliary muscle is relaxed, the zonule are stretched because there is a wide distance between the lens and the ciliary muscle. So when the zonule are stretched, then the lens are flattened, or the lens is flattened. So the lens doesn't exert any accommodation. Again, when the eye is at rest, the ciliary muscle is relaxed and the zonule are stretched so that the lens is flattened. But when the eye is accommodating, when it's seen a near target, look what happens. This is what happens. The ciliary muscle contract so that the zonule are slackened, or by slackening, as I said before, this means that they become relaxed. So that since the lens has an elastic capsule and the plastic substance, so this elastic capsule will stretch, as there is no traction over it exerted by the zonule anymore. So it will stretch like this, leading to increasing the anterior-posterior diameter of the lens, leading to a bulge of the lens, increasing its refractive power, and become more spherical, and that the accommodation is now occur. So this is one theory for the accommodation and how does it occur. The second, let's say here is the accommodated lens, so that you can see that the ciliary muscle will be relaxed, so the zonule are stretched and leading to flattening of the lens. When the lens is accommodating by Helmholtz theory, then the ciliary muscle will be contracting, leading to decrease the ring or decrease the size of the ciliary muscle ring, so that the distance between the ciliary muscle and the lens will be decreased, causing slackening and relaxation of the zonule, leading to that the lens will bulge anterior and posteriorly, causing the accommodation. The third theory is called the Skakar theory. Let's see what is the Skakar theory. When there is an accommodation, there is an equatorial zonular tension is increased, meaning that this will be the reverse of the Helmholtz theory. So Skakar and sometimes also Tischring have also suggested the same theory, that when there is contraction of the ciliary muscle, this will lead to contraction of the zonule. But this contraction of the zonule are in the this will be the equatorial zonular tension is increased because the anterior and posterior zonule are simultaneously relaxed. So here the anterior and posterior zonule are relaxing, meaning that they will allow the lens, the central part of the lens to contract and the peripheral part of the lens to be flattened. Again, the front part and the peripheral part of the lens will relax. So the front zonule and the back zonule will relax. This will lead to that the central part of the lens will bulge and the peripheral part of the lens will flatten. This is called the Skakar theory. Again, so unlike that, the Helmholtz theory, the central zonule become tight here and there, and the peripheral zonule, like anterior and posterior, become relaxed, so that this central part or central zonule that become tight will causing the central part of the lens to bulge, and this peripheral zonule, which are anterior and posterior, become more relaxed, so that it will cause flattening of the lens periphery, and that's what makes the lens assume this globular shape. So this is called the accommodation lens by Skakar theory. And it was found that this theory may explain that the presbyopia occurs due to growth in the equatorial diameter of the lens. It will explain that why the presbyope will develop presbyopia, why they are not able to see the near target, because of the growth of the equatorial diameter of the lens with age. So this will decrease the space between the lens and the ciliary muscle, so that the ciliary muscle cannot contract and cannot cause any tension in the zonule. So the lens, as it grow from the equator, like here and here, this will lead to relaxation of the anterior and the posterior zonule. So here is an example for the accommodation. When looking at the distance, then there will be a relaxation of the ciliary muscle, so the zonule will be tense. If there is looking to a near target, there will be contraction for the ciliary muscle, and this will lead to slackening of the zonule, causing anteroposterior bulge of the lens. Then the ciliary muscle, you can consider it like two persons pushing the lens, so that this will lead to bulge of the anteroposterior diameter of the lens, increasing its refractive power. As we get older, then the ciliary muscle become weak, and so that the ciliary muscle is no longer be able to contract, and so that this will be the lens become we are not able to increase the anteroposterior diameter of the lens, so that this will lead to presbyopia. So to summarize, or to finish, what are the ocular changes that occurs with accommodation? The first of all, as the lens enlarges in size, there will be slackening of the zonule, so they will be relaxed. There will be change in the anterior curvature of the lens. There will be change in the anterior curvature of the lens as it will become more bulging. So the radius of curvature decreases from 11 millimeter to 6 millimeter, as the radius of curvature will be inversely proportionate to the diameter of the lens, or to the surface curvature. So the more the surface curvature, the less radius of curvature. The anterior pole of the lens will move forward, and there will be increasing in the axial thickness of the lens. There will be constriction of the pupil as a part of the near reflex, so the near reflex will consist of convergence of the pupil and and convergence, meiosis of the pupil, convergence of both eyes, and accommodation. So when looking to a near target, this will be the triad that will occur. Choroid is stretched forward, and so is the ora serrata, which moves forward by about 0.5 millimeter. This has been also asked in the MCQ before. What happens occurred to the choroid and the ora serrata? They are moving forward. So during accommodation, they are moving forward by about 0.05 millimeters. So this is the end of the lens lecture. Do you have any questions in the lens and the accommodation? Okay, we will stop now by this amount. So we will not take extra lectures today. I don't want to make it excess for you, as long as many of you didn't have the opportunity to sit for the optics exam, and you are probably busy with the studying of the optics. So I don't want to overload you. So this will be the end of our lectures today. And we will carry on with the group. So we will say, or we will tell what will be the schedule for the remaining lectures. Okay, so reduce the glutathione. There is a question asking about what is the importance of the reduced glutathione in the eye. So let's say that this is the glutathione. When the glutathione is combined with hydrogen, and there is here in the lens, there is an oxygen or oxygen free radical. So when this compound of reduced glutathione combined with oxygen, this oxygen will bind with this hydrogen to form the H2O. So now this reduced glutathione was able to protecting the lens from the damaging effect of this oxygen free radical. But if this glutathione is not reduced, it will not be able to combine with oxygen, so it will be this oxygen free radicals will accumulate within the lens. You are welcome. So thank you very much for your attention, and thanks for those who attended and sat for the optics exam. And I wish you all the good of luck and a good night.